Hello viewers, welcome you all for our lecture series on agrochemicals and pest control. In today's episode, we are going to discuss about the topic host plant resistance. The term host is used to indicate a plant species on which a plant parasitic species can feed and reproduce. The general condition, however, is that a given plant species is a non-host to most nematode species. That is, most nematode species do not use the plant as a food source. The term resistance is used in a non-epidemiological sense to describe the effect of a plant on nematode reproduction. A highly resistant plant allows little or no nematode reproduction, whereas a susceptible plant supports abundant reproduction. Partially or moderately resistant plants permit intermediate levels of reproduction. Host plant resistance pertains to a plant's ability to resist damaging insect invasions. Some plants use their physical appearance as a deterrent, such as plants that have hair covering their leaves or plants with a thick leaf cuticle. With biotechnology came a new form of host plant resistance, the ability to insert genes into plants, creating transgenic hybrids gave a whole new meaning to plant resistance. Plants that previously had no resistance mechanisms now are able to resist damaging insects. Hence, in this episode, the following aspects are going to be seen. They are types of resistance, source of resistance and mechanisms of resistance, methods of breeding for disease resistance, advantages to the use of insect resistant crop varieties, and advantages and disadvantages of host plant resistance. Now, we shall move into the first aspect, types of resistance. Plants show normally two types of resistance to pathogens. Firstly, ecological resistance or pseudo-resistance. Apparent resistance resulting from transitory characters in potentially susceptible host plants due to environmental conditions. Pseudo-resistance may be classified into three categories. They are host evasion. Host may pass through the most susceptible stage quickly or at a time when insects are less or evade injury by early maturing. This pertains to the whole population of the host plant. Next is induced resistance. Increase in resistance temporarily as a result of some changed conditions of plants or environment, such as change in the amount of water or nutrient status of the soil. Next is escape. Absence of infestation or injury to the host plant due to transitory process like incomplete infestation. This pertains to few individuals of the host. Second type is genetic resistance. True resistance or genetic resistance is that in which the plant exhibit resistance because they possess genes for resistance directed against the virulent genes of the pathogen. In this resistance, the host and the pathogen are more or less incompatible with each other due to lack of chemical recognition between them, or the host defends itself against the pathogen by means of various defense mechanisms either pre-existing or developed in response to pathogen attack. Based on the number of genes, host plant resistance are divided into 5 types. They are monogenic resistance. They are controlled by single gene. They are easy to incorporate into plants by breeding and easy to break also. Next is oligogenic resistance. It is controlled by few genes called as oligogenic resistance. Next is polygenic resistance. It is controlled by many genes called as polygenic resistance. Next is major gene resistance. It is controlled by one or few major genes are also called as vertical resistance. Next is minor gene resistance. It is controlled by many minor genes. The cumulative effect of minor genes is called adult resistant or mature resistance or field resistance, and it is also called as horizontal resistance. Next is based on biotype reaction, it is divided into two types. They are vertical resistance and horizontal resistance. Moving to the vertical resistance, it is effective against specific biotypes, also called as specific resistance. Horizontal resistance is effective against all the known biotypes, and it is also called as non-specific resistance. Based on population or line concept, it is divided into two types. They are pure line resistance and multi-line resistance. Pure line resistance is exhibited by lines which are phenotypically and genotypically similar, whereas multi-line resistance is exhibited by lines which are phenotypically similar but genotypically dissimilar. Next is miscellaneous categories. Cross resistance, variety with resistance incorporated against a primary pest confers resistance to another insect. Next is multiple resistance, resistance incorporated in a variety against different environmental stresses like insects, diseases, nematodes, heat, drought, cold, etc. Based on evolutionary concept, it is divided into two types. They are sympatric resistance and allopatric resistance. Sympatric resistance is acquired by coevolution of plant and insect gene for gene, and it is governed by major genes. Whereas allopatric resistance, it is not by coevolution of plant and insects, but it is governed by many genes. Now we shall move into the second aspect, sources of resistance and mechanism of resistance. Three key potential sources of resistance are available, and with them come the obvious requisites of fundamental research to develop their potential. Firstly, wild plant species. Wild plant species represents the most important source of HPR genes, that is host plant resistant genes. Thus far, and no doubt hold in their gene pools considerable additional and as yet unrecognized sources of resistance. However, they present a challenge in breeding work due to problems of incompatibility, particularly among the more divergent taxa or genotypes, and the association of resistance with various undesirable traits. Embryo rescue and somatic hybridization techniques may facilitate otherwise difficult gene transfers. Second is induced mutants. Some mutants induced by irradiation express increased level of resistance to nematodes. For example, in chickpea and potato, although their stability over time is questionable and as yet unknown. Third is plant regeneration. The regeneration of plants from organs, tissues and cells facilitates selection of somoconal variants with desirable resistant traits arising from single nuclear changes. They are preferred over mutation breeding as they retain the desirable characteristics of the existing cultivar. These tissue culture induced genetic variations and their potential as sources of resistance to diseases and nematodes have been little explored in research programs and warranted further investigations. Likewise, the application of in vitro tissue culture techniques, protoplast fusion, etc. towards the routine or special transfer of HPR genes across sexually incompatible boundaries between plants has not been given much research. Next is mechanism of resistance. The three important mechanisms of resistance are antizoonosis, antibiosis and tolerance. First is antizoonosis. It is also known as non-preference. Host plants that express non-preference affect the way an insect pest perceives the desirability of the host plant. Non-preference plants either provide stimuli that are unattractive to the pest color, odor and texture such as downy hairs, or fail to provide stimuli that are attractive to the pest. In this way, non-preference plants affect the behavior of pest. Next is antibiosis. Antibiosis is a type of resistance in which the host plant causes injury, death, reduced longevity or reduced reproduction of the pest. Often both a resistant and susceptible variety will have the same basic responses to a pest, but the resistant variety will respond more quickly or more drastically than the susceptible variety, reducing the amount of damage the pest causes. Plants that express antibiosis affect the biology of pest. Next is tolerance. It is resistance in which a plant is able to withstand or recover from damage caused by insect pest abundance equal to that damaging a plant without resistant characters. Tolerance is a plant response to an insect pest. Thus, tolerance resistance differs from antibiosis and antizoonosis resistance in how it affects the insect-plant relationship. Antibiosis and antizoonosis resistance cause an insect response when the insect attempts to use the resistant plant for food, oviposition and shelter. Now, we shall move into the third aspect, methods of breeding for disease resistance. The methods of breeding varieties resistant to diseases do not differ greatly from those adopted for other characters. The following methods are used. They are introduction, selection, hybridization followed by selection, backcross method, induced mutagenesis, development of multi-lines and tissue culture techniques. First is introduction. It is a very simple and inexpensive method. Varieties resistant to a particular disease elsewhere may be thoroughly tested in the region in which they are proposed to be introduced. Their yield performance and disease resistance should be confirmed by the large scale cultivation. It is possible that a variety resistant in one region need not be resistant in another region due to variation in the physiological race of the pathogen or due to a much different agroclimatic conditions in the new location. Introductions have served as a useful method of disease control. Next is selection. This is better method than introduction and has more chances of success in obtaining disease resistant plants. The work of selection is carried out either in the naturally infected fields under the field conditions or artificially inoculated conditions. The resistance in such individuals will occur in nature by mutations. To ensure the resistant character of a plant, large population of crop plant may be exposed to the attack of pathogen under artificial conditions and the non-infected plants may be chosen. Suvarnamodhan rice of Kerala is a pure line of ARC 11775 and it is highly tolerant to blast. Next is hybridization. When selection of resistant varieties is not feasible, resistant varieties may be evolved by crossing the susceptible popular variety with resistant wild variety wherein the resistant gene or genes transferred into the genetic makeup of susceptible variety. Very often the F1 from crosses may be resistant but carry the other undesirable qualities of the resistant parent. The bad qualities are removed by several back crossing of F1 with the susceptible parent and may ultimately yield a resistant progeny with good agronomic characteristics. Under certain circumstances, pedigree or bulk method of selection is followed to obtain a resistant variety. In this method, the crosses are made till F2 population is got. Selections are made in F2 generation for superior genetic traits including disease resistance. Selections are made through F3 to F5 or F6 generations and the best variety is selected. This method is suited for small grains and beans but unsuited to fruits and vegetables. Next is back cross method. Back cross method is widely used to transfer disease resistance from wild species. Wild species are rice sources of disease resistance. Interspecific hybridization is made to transfer the gene or genes of resistance to the cultivated species. Resistance to grassy stunt virus from Oryza nivara to Oryza sativa, late blight resistance from Solanum dimysum to cultivated potato, rust resistance from durum to aestivum wheat are some of the examples involving interspecific hybridization. Depending on the number of genes governing resistance and the nature of the genes, whether dominant or recessive, the procedure varies. The number of back crosses to the cultivated species may be 5 to 6. Once the back cross progeny resemble the cultivated parent and segregating progeny screened for disease resistance. Next is induced mutagenesis. The mutation breeding for disease resistant, a large number of mutation progeny should be produced and screened under artificial epiphytotic conditions to select resistant plants. Next is development of multilines. The concept of multilines was first suggested by Jensen in 1952 and was developed by Borlaug in 1959 for evolving multiline varieties to resist stem rust in wheat. A multiline variety is a composite of genetically similar lines except that each line possesses a different gene for resistance to the pathogen. Lines that are genetically similar except for one gene are called isoline. It is assumed that gene for resistance in each isoline contributes resistance to a separate physiological race or group of races. Genes for disease resistance are transferred by backcrossing from donor varieties to a common disease susceptible but agronomically superior recurrent parent. Isolines are generated differing only in the gene for disease resistance. The first multi-line variety in wheat, that is Miramar 60, was developed and released in Colombia to combat the attack of yellow rust. Miramar 63 and Miramar 65 were resistant to stem rust and stripe rust. Yoku 50, Crew and Tumboldt are the few other wheat multi-lines. Kalyan Sona and Sonalika based multi-lines of wheat resistant to different races of rust have been developed in India. Next is tissue culture techniques. Tissue culture techniques to produce somoclonal variations for disease are developed in different crops. Somoclonal variations for disease resistance are reported in Zea mays for Dicrocera myadis race T toxin resistance, in Brassica napus for resistance to Phoma lingam, early and late blight resistance in potato, Pseudomonas and Alternaria resistance in tobacco, besides smut and rust disease resistance in sugarcane. Use of insect resistant crop varieties is economically, ecologically and environmentally advantageous. Economic benefits occur because crop yields are saved from loss to insect pest and money is saved by not applying insecticide that would have been applied to susceptible varieties. In most cases, seed is insect resistant cultivars cost no more or little more than for susceptible cultivars. Ecological and environmental benefits arise from increases in species diversity in the agro ecosystem, in part because of reduced use of insecticides. Increase in species diversity increases ecosystem stability, which promotes a more sustainable system, far less polluted and detrimental to natural resources. The integrated pest management concept stresses the need to use multiple tactics to maintain insect pest abundance and damage below levels of economic significance. Thus, a major advantage to the use of insect resistant crop varieties as a component of integrated pest management arises from the ecological compatibility and compatibility with other direct control tactics. Insect resistant cultivars synergizes the effect of natural, biological and cultural insect pest suppression tactics. The built-in protection of resistant plants from insect pest functioning at a very basic level, disrupting the normal association of the insect pest with its host plant. The compatible complementary role plant resistant to insect pest play with other direct control tactics is in the theory and practice in consent with the objectives of integrated pest management. All crop cultivars should contain resistance to insect pest. Plant resistance to insect pest have advantages over other direct control tactics. For example, plant resistance to insect is compatible with insecticide use while biological control is not. Plant resistance to insects is not density dependent, whereas biological control is. Plant resistance is specific, only affecting the target pest. Often, effects of use of insect resistant cultivars are cumulative over time. Usually, the effectiveness of resistant cultivar is long lasting. The role of plant resistance to insect in integrated pest management has been well defined, at least in the theory. However, the specific role a resistant cultivar plays in the particular integrated pest management situation is crucial to successful deployment of the resistant cultivar. The impact of the resistant cultivar on standard cultural, biological and insecticidal control methods should be well defined. Likewise, the impact of each of these control tactics on the resistant cultivar also must be defined. Advantages of host plant resistance as a component in integrated pest management are specificity, specific to the target pest, and natural enemies are unaffected. Next is cumulative effect. It lasts for many successive generations. Next is eco-friendly. No pollution, no effect on man and animals. Next is easily adoptable. High yielding insect resistant varieties easily accepted and adopted by farmers, and they are less cost. Next is effectiveness. Resistant variety increases efficiency of insecticide and natural enemies. Next is compatibility. HPR genes, or otherwise called as host plant resistant genes, can be combined with all other components of integrated pest management. Next is decreased pesticide application. Resistant varieties requires less frequent and less doses of insecticide. Next is persistence. Some varieties have durable resistance for long periods. Next is unique situations. HPR genes effective where other control measures are less effective. For example, while timing of application is critical, crop of low economic value, pest is continuously present and is a single limiting factor. Next comes disadvantages of host plant resistance. Time consuming. It requires from 3 to 10 years by traditional breeding programs to develop a resistance variety. Next is biotype development. A biotype is a new population capable of damaging and surviving on plant previously resistant to other population of same species. Next is genetic limitation. Absence of resistant genes among available germination. Now, we shall conclude this topic. The ability to develop insect resistant cultivars use a marker assisted selection and development of transgenic plants with insect resistance depends on the precision of resistant screening techniques. So, students, hope you enjoyed this session. We will meet in the next episode. Until then, take care. Thank you.